Now we'll talk about galactose operon, which is one of the best studied catabolite sensitive operon. It contains 4 structural genes, that is GAL-K, GAL-T, GAL-E and GAL-M, and these 4 structural genes, they specify 4 enzymes, that is galactokinase, galactose transferase, galactose epimerase and mutarotase. Mutarotase converts the beta D galactose into alpha D galactose. Beta D galactose कहाँ से आया? Beta D galactose is formed when beta galactosidase acts on the lactose and converts lactose into galactose and glucose. So as a result of this reaction, beta D galactose is formed, and then this mutarotase converts this beta D galactose into alpha D galactose. Then galactokinase, galactose transferase and epimerase, these 3 enzymes act in a sequence of steps to yield the overall reaction, and the overall reaction is galactose plus ATP, they are converted into glucose 1-phosphate plus ADP. So this is overall reaction of these 4 enzymes. The galactose or GAL operon, like the LAC operon, is regulated by a repressor and CAMP-CRP complex. The structural genes for the repressor GAL-R is located far from the structural genes for the galactose enzymes. The GAL operon also has 2 operators, that is GAL-OE and GAL-OI, a promoter region and a CAMP-CRP activator site. So ये सब चीजें होती हैं एक GAL operon में. The promoter regions contain 2 promoters, that is P1 and P2. तो ये पूरा स्ट्रक्चर है जीन इसका galactose operon का. आप इसमें देख सकते हैं this is GAL-E, GAL-T, GAL-K and GAL-M. So GAL-E makes epimerase, GAL-T makes galactose transferase, GAL-K makes galactose kinase and GAL-M makes mutarotase. So these 4 enzymes are formed. This is operator GAL-OI. This is another operator GAL-OE. This is the activator site, and these 2 are promoters P1 and P2. So GAL-R can bind here, here and here, and CAMP-CRP complex can bind here, and this is a histone-like protein, and it can bind here. So this is the structure of whole galactose operon. One operator's GAL-OE is upstream from the promoter region, and the other GAL-OI is in the GAL-E. A mutation in either operator causes at least partial constitutivity, indicating that both operators participate in repression. The GAL repressor, which binds to both operators, is made up of 2 identical polypeptide chains. A null mutation in GAL-R makes both promoters constitutive. The amino acid sequence of GAL repressor is similar to that of the LAC repressor. There is one important difference, however, and that difference is the GAL repressor doesn't have the C-terminal residues that allow the LAC repressor to form a 4 helical bundle and become a stable tetramer. So this is the difference between GAL repressor and LAC repressor. The GAL repressor has a C-terminal domain that binds the inducer galactose and an NTD or N-terminal domain with a helix-turn-helix motif. Helix-turn-helix motif means alpha helix, then beta turn, and then alpha helix. These terms are used in protein structures. So then it has this motif that binds to a GAL OE or GAL OI half site. The GAL repressor is a dimer and not a tetramer, and the studies which indicated that both GAL OE and GAL OI are required for repression suggested some type of cooperative interaction. Sankar Adia and co-workers solved the problem by showing that a histone-like protein called HU binds to the GAL promoter region only when the GAL repressors are bound at both the GAL OE and or GAL OI, causing the DNA to loop. So here you can see this is GAL repressor dimer. ये डाइमर है और ये जो है ये GAL OI के साथ बाइंड है. इसी तरह ये जो है ये भी डाइमर है और ये GAL OE के साथ बाइंड है. दिस इज DNA और यहाँ पे ये जो है हिस्टोन लाइक प्रोटीन है, एंड दिस हिस्टोन लाइक प्रोटीन causes the DNA to loop, and this is the promoter region. The two GAL promoters P1 and P2 are separated by 5 base pair, and as a result the transcription initiation site for P1 is 5 base pair upstream from that of P2, क्योंकि इनमें डिफरेंस जो है वो 5 बेस पेयर्स का है.